class. Final abstract due tomorrow. This is Cancer Immunology 2 and 3. So these are second and third year students, primarily juniors and seniors. They're working on different projects that were developed during the first year. And some of them have continued it all the way on to their senior year. Projects ranging from using CRISPR gene editing system to disable genes that we think are associated with cancer. And then we check for phenotypes. Does it stop the cells from growing? Does it kill the cancer cells? There's also students working with treatments. And then we also have students that work strictly with computational biology, where they're analyzing genes and trying to find gene mutations or gene patterns that are associated with cancer.
So if you look closely, down here, those are all my cells. They're like in like a pellet at the bottom.
So we're holding our streaked plates, and it's basically like bacteria that we diluted to form isolated colonies. And today we're going to be doing a mini prep with these.
These cells weren't growing confluently. We're going to aspirate them and get rid of them, and we started a new culture today.
I'm going to be diluting cells today because I'm growing glioblastoma cells and they're really fast at growing. So we're melting TE. These are going to need like a couple minutes to melt.
I'm cryopreserving my cells today. My flask had about 90% confluency, which is pretty high. So I'm going to be cryopreserving these two. I just made cryoprotected medium, which is DMSO mixed with my regular medium. And then I'm going to take these two, which have about 1 milliliter each of my cells mixed with the medium. I'm going to put them in the negative 80-degree freezer and then move them to the liquid nitrogen tomorrow. And then I'll be able to thaw these if I ever need to grow more cells.
This is a leukemia cell line derived from a male patient. I'm not sure the age. The cells, they move a lot because they don't stick to the bottom until a day of being in the incubator.
Would you be concerned why it's heavy?
No, that's the old TE that had a little bit of phenol red in it. When I was working on my doctorate at the Cleveland Clinic, as teachers, we all have afternoon commitments. And so my afternoon commitment was to take students with me to the Cleveland Clinic where I would get to work on my research and then they would help me out. And so they would get that training opportunity. That flourished into internships. And it was during that internship that I got to see just how much responsibility the students can take on. All the other scientists that the students were helping were very impressed with them as well. That's where the idea was born, because I was about to complete my doctorate. Back then I was teaching AP biology. You don't need a doctorate for AP biology. So I said, what am I going to do with this knowledge? And so the idea spawned, maybe I can start a research program there at Western Reserve Academy. And so little by little, I started gathering all of the different instruments that you see throughout the lab. Through that, donations from CASE, from the Cleveland Clinic, and also the Pioneer Women's and the Dads Club, I managed to put all of this equipment together and bring this experience to the students.